Hey folks, we're back going to talk a little bit more about the nervous system, and today our concern would be on the meninges and the cerebrospinal fluid and the spinal, and a little bit about the spinal cord with the spinal fluid that's around it. I guess you could probably tell that the coronavirus virus has really me a little bit frazzled, so hopefully I'll be able to finish this out. Hang on, just a minute, just a minute. Okay, I'm back. I think we're better now. So let's just talk a little about this about the meninges, which are basically the membranes that cover the brain, as well as the fluid that is produced within the brain. And I'll talk a little about what the purpose of that is and how it's made and where it goes and stuff like that in just a second. So let's talk about that. Let's first talk about the meninges. Okay, the meninges are the membranes that actually cover the brain, and there are three membranes. Okay, a couple layers to each, but I won't get into too much about the layers of those. But there are three basically membranes that cover the brain. The first and the outermost layer is called the dura matter. Okay, and the dura means durable, means very hard, and it's very thick. It's very rough. When they actually do an autopsy and they have to take the top of the skull off and then pry the skull off to be able to look at the brain, they actually have to sort of shell the dura away from the inside because the dura matter is really very tough. It's very fibrous. It's mostly really dense connective tissue. It's the outer layer and it's stuck to the inside of the cranium, inside of the skull. Okay, so you have to peel it off the inside of the skull to be able to get the top of the skull off once they cut around the skull, and that's called the dura. Okay, and you've probably heard of subdural, epidural, and I'll explain a little about that in a second. The second layer that we have is called the arachnoid layer. And the arachnoid layer is attached to the undersurface or the inside surface of the dura. Okay, so the dura is actually two layers thick, and then the arachnoid is attached to the bottom of the dura. Okay, it's called arachnoid because underneath the arachnoid membrane, there's a larger area or a gap, and it's filled with all these little spidery web-like fingers that come from the arachnoid that go down to the third membrane, which is called the pia. The brain, I'll show you a little diagram of that. And the spidery nature of the membrane as it weaves through this space, it gives a great name called the arachnoid layer. Like arachnid spider, so arachnoid layer. The third layer of the meninges that we have is called the pia. The pia is actually a very thin membrane, and pia means delicate, fine, thin, delicate, and it's actually stuck to the outside surface of the brain. So last time we talked about the gyri and the sulci and stuff like that. What happens is it looks like brain tissue, but it's still covered with this membrane, and this membrane would be called the pia. Now, going back to that significance of that gap between the arachnoid and the pia, again, you remember the arachnoid is attached to the undersurface of the dura, and the pia is attached to the surface of the brain. But between the arachnoid and the pia, there's a small space with all these little web-like fingers, okay? So there's the space between the pia on the surface of the brain and the arachnoid membrane with these little web-like strings, and that's called the subarachnoid space. It's called the subarachnoid space. Sub means below, arachnoid, and it's a space, okay? The real important part about what this subarachnoid space is that's where the cerebral spinal fluid is. Well, we haven't talked much about cerebral spinal fluid, so we don't know, but that's where it's at. So we'll talk a little about that, about that membrane. Now, if we look at the brain, this is just a diagram of the brain. Here'd be the brain right here. It's sort of hard to see, but the skull would be the outer portion out in here, this area, and then the dura would be in there. Okay, so the dura is attached to that, and then what happens is the arachnoid is just under the dura, and then the pia sits on the top of the brain, and there's that subarachnoid space. A little bit better picture, might be looking at the spinal cord. Now, let me mention what happens. These membranes, once they cover the brain, the dura, the arachnoid, and the pia, all of them will continue down out through that foramen magnum, which you should know, or you better know, out through the foramen magnum with the spinal cord, and they cover the spinal cord the same way. So this outer layer in here would be the dura. Okay, so that's the dura that's around the outside, very thick, very dense, and then attached to the inside of the dura would be the arachnoid, which leaves this space right here, and that space right there would be the subarachnoid space. Subarachnoid space. And guess what's in there? That's cerebral spinal fluid, like we talked about. What's on the surface of this is the spinal cord right here, and what's on the surface of the spinal cord? The pia. The pia is attached to that. So my dura is on the outside, okay, and it sort of lines that vertebral or spinal canal. The arachnoid is attached to the undersurface of the dura, and then there's a gap with all these little web-like fingers. It's filled with fluid called the subarachnoid space, and then the pia would be on top of the spinal cord itself. Okay, and this is just another little diagram that shows that this outer yellow layer out here would be the dura, okay, and the arachnoid is attached to the undersurface of that, and then the pia is sitting on the surface of the spinal cord, and then right in here between right in this area right here between the pia on the spinal cord and the arachnoid, which is attached to the undersurface of the dura, is that subarachnoid space where that's spinal fluid. So basically, if I'm looking at this, let me get rid of this stuff here. If I look at this, if you look at the gap right in here, this gap would be where that gap would be, that subarachnoid space is, because this membrane right here is the arachnoid. Here's the dura, and they're attached, and this membrane right here would be the pia. Okay. If I'm looking at the inside of the skull, this is probably a little bit better diagram than you might see. So this is the inside of the skull. Okay, that's that, you know, flat bone, you know, with a cortical shell on the inside and the outside and cancers in the middle, and then right on the inside surface attached to it, just doesn't show it as attached to it as well as it should, but this light blue layer right here would be the dura. It's actually attached to the inside of the skull. Okay, and then what happens is this next layer right here, it's a little bit whiter, is the arachnoid, which is attached to the dura. These are all those little webs. If you look at here, these are all those little webs, and that fills this space inside here. Let me just do it with a different color. Let's do it with light green. That looks good. Look at fill in this area. It's not going to show anyway. Anyway, this area in here is that subarachnoid space, and then right on the surface of the brain, right along here is where we see my pia that's attached. Actually, we'll go down all the way in all the sulci. It covers the surface of the brain really close. If you ever go and get a brain from like the supermarket or for the market or you know West Side Market or something like that, you grab the surface of a cow brain or whatever, and you with a pair of tweezers or pickups, and you pull up, you'll pull up a little membrane, and that would be the pia. Okay, and this also shows the same thing. This is just another diagram. So we see the yellow up in here would be the skull. Okay, the dura is sort of like this yellowish greenish. Okay, the arachnoid is attached to the bottom of the dura. This lime green would be called the subarachnoid space, and that's where the spinal fluid is on, and then this very thin red line in here would be the pia. So basically that's a pretty good diagram of showing what we have. One last thing I just want to mention, this shows it really well, is again what we're looking at here. Here's this upper layer in here. Let me just do it this way. Okay, the upper layer in here is the dura. This layer right here is the dura. Okay, if I look down in here, this next layer right there is the arachnoid. Here's the subarachnoid space. This area in here is that subarachnoid space. Okay, and then the pia is sitting right here on the surface of the brain. Now, if you look, there's all these little webs, and that's where that cerebrospinal fluid is going to be. Now, here's the key. What happens is when we talked about circle of Willis a little bit in the lab, it's a circle, a vascular circle on the bottom of the brain. We'll talk about that when we get to the cardiovascular section. It's on the bottom of the brain that supplies the circulation to the rest of the brain. It's a circle, like a driving circle, a traffic circle, has all these branches coming off that supply the rest of the brain. What happens is most of these large branches that come off of that circle of Willis sit on the surface of the brain like that. So if you took a brain out, you see all these big arteries on the outside of the brain, and so what happens is if somebody has a cerebral hemorrhage, or what's called a subarachnoid hemorrhage, again below the arachnoid, so it sits in here, blood is going to fill this space. So when they do a lumbar puncture, because that space is continuous all the way down through the spinal cord, when they put a tap in there, the spinal fluid, which is normally water, looks like water, it's clear, is going to be either tinted sort of a yellowish color or will be grossly bloody. Okay, and I'll show you that in a second here. Okay, so what is that cerebral spinal fluid for? And cerebrospinal fluid is mostly used for fluid protection. In other words, it provides a little bit of a buffer, so when the head gets rattled around, the head has a little bit of a fluidy bag to sit. Now it's not much, okay, just a very small, but does allow a little bit. Plus, it also provides some nourishment, you know, to the area of the brain, the surface of the brain. So it bathes the brain and the spinal cord because, again, it's continuous all the way around the brain and down around the spinal cord. When we do a lumbar puncture or a spinal tap, what they do is they take a needle, I'll show you a diagram of that in a second, they take a needle in the lower lumbar region, and I'll explain that in a second, and they just draw the fluid out. Again, that cerebrospinal fluid is normally colorless. It comes out, looks like water. They measure the pressure of it, how much pressure is in the of the fluid, and what's in. Then they take about four tubes, and some of them they just look for gross appearance, some they look for cells, some they look for bacteria, and then one they keep for later on just to see if they need it for anything else. And the cerebrospinal fluid does carry a number of things like oxygen. Whoops, I didn't want to blot that out. Oh, that was bad. Okay, I should have called that differently. Let's go this. Let's do this. It supplies, carries oxygen, glucose, and other needed chemicals from the blood to neurons and the neuroglia, okay, to provide nourishment. In fact, what happens in bacterial infections, not only will the cerebrospinal fluid become sort of cloudy and turbid with pus in it, but when they measure the chemicals, the glucose level, which is needed for nutrition, goes down. Why? Because the bacteria eating all the glucose, okay? And the protein level in the cerebrospinal fluid goes up. Why? Because we have a lot of cells and other garbage in there, and inflammatory proteins that are produced inside there. So they can actually sometimes look at these chemicals on the cerebrospinal fluid, on the spinal tap, to be able to determine what might be going on with the brain as well, besides just the color. Okay, and again, this circulates, and eventually it's absorbed. It's absorbed in a very interesting way. And I'll mention that when we get to the very last slide to talk about something at the very last, okay? And this is what the cerebrospinal fluid will look like. Again, normally it's just very clear. It looks like water. However, sometimes, and this is called xanthochromic. Xantho means yellow, and it's like a yellow tinge to it. And sometimes it's yellow tinge because there's a little bit of blood, and that little bit of blood that gets in there tinges it. However, if there's lots of gross bleeding, this area will be much more bloody. If there's an infection, this area you can't see through. It's not looking like water. It looks like almost like milk. It's very, very turbid. Okay, and so they could actually look at that. And the way they do that is they actually come down here between the third and the fourth lumbar vertebrae. Okay, and then they just put the person in a side position just like they have here. Knees would be up and These would be up in here. This would be the back, and they curve the back this way. And that opens that gap between those spinous processes of the vertebrae. And again, the reason why they go between the third and the fourth, as we talked about this a little bit in the lab, is that what happens is the hard part of the spinal cord actually stops just a little bit above that. And below that level, we have that cauda equina, a series of strands of nerves that come down that will then leave out through all those holes between the vertebrae and as well as in the sacrum to go where they need to go. So as a result, the chance of actually taking a needle and sticking it through the spinal cord when they go between L3 and L4, between the third and fourth lumbar vertebrae, is very low. Simply because it's like hitting a noodle. You have a little strand of spaghetti in there as compared to a solid spinal cord structure. Okay, so that's what they do with a spinal tap, or it's actually medically called lumbar puncture. Okay. Now, so now we know that there's something called cerebrospinal fluid, and we know that it's in that subarachnoid space, which is between the arachnoid and the pia, and the arachnoid we know is attached to the undersurface of the dura. Duh, we got all that already. How is this cerebrospinal fluid produced? Well, it's produced and contained in what are called ventricles. When people say ventricles, they automatically think of the heart, but what happens to our ventricles in the brain? We have two lateral ventricles, and they're almost like C-shaped open areas in the area of the cerebral cortex, okay, and they're sort of C-shaped. Now, inside these ventricles, there's special cells. It's called the choroid plexus, and this choroid plexus has cells that are actually able to make the cerebrospinal fluid. Okay, what happens is there's a lateral ventricle on the right, a lateral ventricle on the left, and what they do is they eventually make this fluid and drains to the middle. It goes to what's called the foramen of Monroe, but I don't want you to actually remember that, but it goes to what's called this midline or third ventricle. So between the lateral ventricles and down a little bit, there's a third ventricle. And as a result, it just sits there. I'll show you where it would be in an actual diagram of what the brain looks like. What happens is then from the third ventricle, it goes down through another canal and goes into what's called the fourth ventricle. Okay, and the fourth ventricle is close to the pons and the medulla. Okay, and from this fourth ventricle, there are other channels, so like 3 channels that come out of that fourth ventricle that will then go and surround the whole area of the brain to provide the cerebrospinal fluid around the whole area of the brain, as well as the cerebrospinal fluid going down the spinal cord. Again, always in the subarachnoid space. Okay. So it's always in that subarachnoid space. Now, if I look at the brain, this is what we see. Let me get rid of my little markings here. get rid of that stuff, get rid of that stuff. And this is what we see, okay? And basically, let me just go and mark it up again. Here's a lateral ventricle right here, this C-shaped area, and here's one on the other side. So one will be on the left, one will be on the right. And what happens is this one right here, this foramen of Monroe on the left and the right, feeds into this, well, let me do it a different color. Let's do it to a light blue. Feeds into this area, and that's the third ventricle. So that's the third ventricle. Then the third ventricle feeds down this channel down here to this area down here, which would be called the fourth ventricle. That was a mess, okay? Not too dark. So it feeds down to that fourth ventricle, and from the fourth ventricle, it comes out through multiple channels, 3 or 4 channels. They will then surround the whole brain with the cerebrospinal fluid, as well as around the spinal cord. And that's what that happens with that. So what happens is the ventricular system does hold the cerebrospinal fluid, does hold it. Now, abbreviations for cerebrospinal fluid, we CSF, obviously. And again, it drains into these subarachnoid cisterns around the base of the brain and around the spinal cord and stuff like that. So we know the cerebrospinal fluid, it's hard to say really fast. The cerebrospinal fluid is produced in the choroid plexus in the ventricles. All the ventricles have the ability to produce it, okay? So, and then once it's made, it comes out of this ventricular system and then surrounds the brain and the spinal cord. So now the interesting thing is as it goes around the brain, okay, there are little things, and you don't have to remember this, but I think it's actually sort of cool. I don't want so much of that fluid to build up. In other words, if the fluid's there, we're constantly making fluid. If we're constantly making fluid, we have to constantly be dissipating the fluid or getting rid of the fluid. What happens is in the arachnoid has small little bumps that actually go up into the area of what are called venous sinuses. Now, the dura, I mentioned before, was 2 layers. Sometimes in certain areas those 2 layers will separate. And what that fills, those 2 layers, is venous blood that's going to be heading back to the heart. What happens is these little things are called arachnoid granulations. They actually stick up into that area of the venous sinuses and allow draining off or siphoning off the extra or excess amounts of cerebrospinal fluid. Okay, so basically it's circulating around the brain and down the spinal cord, but the excess is being drained off or siphoned off at these little arachnoid granulations that are all little buds off the arachnoid into these venous sinuses to drain off the extra cerebrospinal fluid. Okay, this is just another diagram. We see these large C-shaped lateral ventricles and stuff like that, and then it goes into a third ventricle that would be in the middle. Okay, third ventricle, it's sort of like you can't see it's down here, and third ventricle was the fourth ventricle. Okay. Now, this is just looking at what we see. If they got a radiograph of the skull, this area right here, you can actually see is the lateral ventricle. You actually see this open area right here. And again, you're only catching one slice of it on this, but it's much more. It's on a CT. This is a CT scan. You can see this larger C-shaped area. What happens is that lateral ventricle is going to drain into this area right here. Let me do it this way. It's going to drain into this area right here, which is basically my third ventricle. And that third ventricle is going to drain into this area right here, which is my fourth ventricle. And that's going to go all the way up around and surround the whole brain and spinal cord in that subarachnoid space. Okay. Now, one thing I just want to mention here, and you don't have probably don't have to remember this, but this is something interesting. Years ago, we used to see a lot of kids with these. When I was a kid, a long time ago, like before there was even writing, I think. What happens? They have these huge heads. Okay, and you still sometimes see that, and it was called hydrocephalus. And what happens is you got to remember those lateral ventricles drain to the third ventricle ventricle drains to the fourth ventricle drains out around in that subarachnoid space around the brain and spinal cord. Sometimes that gets blocked off, or sometimes that duct doesn't form. As a result, the fluid starts to build up, okay, and it builds up. Now this is a normal person right here, and this is the lateral ventricle right here. It's the lateral ventricle right here. Here's the lateral ventricle right here. And that's about the normal size we'd see on this axial plane, because everybody knows what axial plane view would be. On the other hand, if that lateral ventricles can't drain, they keep on filling up with fluid, and all of a sudden now I have this whole area. This black area in here is just all filled with fluid. Okay, and here's another picture. This is all the ventricles that are significantly dilated because they're all filled with the fluid, because the fluid's all dressed up, no place to go. It can't get out, or can't go from the lateral ventricles to the third ventricle, or from the third ventricle to the fourth ventricle, or the fourth ventricle to around the brain in that subarachnoid space. And it's called hydrocephalus. All they do now, when they do decide this, which is that they just can determine it very early, is they actually run a shunt. They stick a tube inside the ventricles and run it down and either empty it into the abdomen or empty it into the jugular vein in the neck or empty it into the heart. And as a result, it gets rid of the extra fluid. Sometimes it has to be pumped or sometimes it clogs up, which is a problem. So it's not a good thing to have, but there are some treatments for it now. The reason why that in the past, the problem was before they knew how to treat it, these kids would have huge heads, and what happens is squeeze the cerebral tissue against the skull, and therefore there'd be significant mental retardation and all kinds of physical and sensory problems that would occur because of that. So that's called the normal hydrocephalus. Now these meningeal spaces we talked about before. A couple of spaces I should mention. Number one, epidural. Epidural is on the outside. Got to remember that dura is attached to the undersurface of the skull. It's stuck there. However, sometimes if that dura rips away and there's bleeding, that's what they call an epidural hematoma. When they do an epidural anesthesia, what they do is they go down right to the area of the dura and inject that area with local anesthetic and surround the area around the nerves that are coming out on the spinal cord with the local anesthetic, and it numbs those nerves. And that would be called an epidural block. Okay, but the epidural space is outside the dura. Subdural space is sometimes between the dura and the arachnoid. Okay, sometimes that dura will actually rip, especially the area of these venous sinuses, which I'll show you about in a little bit, and then it bleeds and actually starts to separate the arachnoid, which is thinner than the dura, from the dura, and that's called a subdural hematoma. Usually it's venous blood, low pressure. It takes a long time to develop. That's when grandma falls and she's okay for a while, but then as the hours go by and the next day she's starting to act a little funny, a little bit goofy, losing more consciousness, sleeping all the time. Next thing you know, you can't wake her up, but she has what's called a subdural hematoma. Now, the subarachnoid space is significant because we talked a little bit before about how the main vessels that supply the brain are on the surface of the brain in that subarachnoid space with that cerebrospinal fluid. Therefore, people who have various strokes, which is called a hemorrhagic stroke, what may happen is one of the vessels that are on the surface of the brain, usually where it branches, there might be a little weak area or a little bubble called an aneurysm. They bust, and when it busts, all of a sudden, next thing you know, we have blood that leaks out. Where's it going to go? In the subarachnoid space. So that's why when they do a lumbar puncture and stick it there might be blood, or the cerebrospinal fluid will look yellowish tinted. Okay, so that's called the subarachnoid space. Okay, and again, we've seen that before, so I don't want to take any more time. We've seen that before. I want to take any more time with that. Let me just tell you one fun fact, the last fun fact about the dura, and I've talked about this before. And what happens is, I mentioned the dura is 2 layers thick. It's very thick. Okay, it's not a vessel, but in cases in the brain, it does serve as a vessel. Okay, we know arteries take blood away from the heart, and they're under high pressure. We know veins bring blood back towards the heart, and they're under low pressure. Okay, so what happens is, in various areas of the brain, that dura that's in 2 layers will actually split and creates a gap between the 2 layers, and that gap is where all the veins will feed into, and it's filled with venous blood. Okay, and these are called dural sinuses. We talked a little bit before about how those arachnoid granulations from the arachnoid membrane with the little bubbles that come up out of the membrane and siphon off the extra cerebrospinal fluid. What happens is that they drain into that dural sinus. Okay, so these dural sinuses are filled with venous blood. So this drains the venous blood to the base of the skull through the jugular foramen. I'll show you how that happens in just a second with a diagram. All these venous sinuses are connected, and what happens, they all go the same way. Remember in lab, we showed about there was one area, like a groove in the posterior cranial fossa, how it sort of like came around like an S shape, which is called the sigmoid sinus. And that actually ended up right at that jugular foramen. And at the outside the jugular foramen, that's where the internal jugular vein starts. So what happens is all the venous blood from the brain collects in these dural sinuses, eventually goes down the sigmoid sinus and out through that jugular foramen, and then that's where the jugular vein starts at the base of the skull and goes down from there. Okay, so that's just sort of a little fun fact about the dura, and hopefully I had a picture, but I'll have to bring in a picture later. Hopefully you understand this. Okay. In summary of what we talked about today is that what happens is we know that the brain is covered by membranes. The outer tough layer is called the dura, 2 layers thick, Sometimes it splits, causing these venous sinuses. The layer that's attached to the undersurface of it is called the arachnoid. It's thinner. And then what happens is that arachnoid has small web-like extensions that attach to a membrane that covers the surface of the brain. And that membrane, that very thin, fine, delicate membrane that covers the surface of the brain is called the pia. And in that subarachnoid space, that gap between the pia and the arachnoid is that where the cerebrospinal fluid is. We know the cerebrospinal fluid, like we talked about before, is made in the ventricles, and the ventricles have tissue or parts of them. They're called the core. You know, that make that cerebrospinal fluid. Okay, it's a choroid that makes the cerebrospinal fluid. Okay, and what happens is that cerebrospinal fluid then goes to the lateral ventricles, to the third ventricle to the fourth ventricle out, and surrounds the brain and spinal cord, providing nutrition, chemicals, glucose, all kinds of other stuff that's needed for the brain and the surface of the brain and things like that. And so that's what we wanted to talk about today. And I put this a little bit out of order. I moved it up simply because I thought it was important because we just finished the cerebral cortex. So hopefully you'll look this over and it'll make a lot of sense to you. If you have any questions, obviously, you can always give me a call or email or whatever, or yell real loud if I'm in the neighborhood.